this lecture we will understand surface tension and its implications in driving flows in microchannels. This is one of the very important aspects of microfluidics, and the reason is that surface tension is a force that acts at the interface, and microfluidic devices have very small volume as compared to a large surface area. So, area by volume ratio is very large, and that makes these devices amenable to manipulation by using surface effects. So, to be brief in telling you what is surface tension, let us say that there is a container. In this container there is a liquid and there is a vapor or a gas at the top. So, if you have some molecule in the liquid, this molecule is being attracted from all its sides by other molecules, and in this process this molecule is in equilibrium. However, if you consider a molecule at the interface, then this molecule is acted upon by different forces. What are the different forces? The different forces are due to the fact that from the top you have the force by the vapor molecules, and from the bottom you have force by the liquid molecules. Liquid being much more dense than vapor, the force of attraction of liquid molecules on the interfacial molecule will be much more, and that will try to bring this molecule towards the inside. But this molecule does not go towards the inside, because if it goes to the inside then the interface will not be stable, but the interface remains its composer. That means the interface has some energy which overcomes this differential force and keeps the interface as it is, and this energy is called as surface energy, and because of this differential force it is therefore can be perceived that the interface is in some kind of tension, and that is called as interfacial tension or surface tension. So, surface tension accordingly is expressed as either force per unit length, that is Newton per meter, or surface energy per unit area. So, that is also Newton per meter. Now, the question is that what can surface tension do in microfluidics? Surface tension is a beautiful force in nature. You will see, think of tall trees. So, when there is a water that is sucked from the ground, the water reaches the topmost branches of the tree without any pump. So, this movement of water, which is called as ascent of sap from the ground to the topmost level, is because of the capillary effect, which is nothing but a surface tension driven flow. So, we will understand in this lecture how that is possible and how that can be applied in microfluidics, but given the significant volume of contents, I will try to go through this using the slides. So, the first point is, how can surface tension actuate and control fluid flow? One is, you can use surface tension as it is. The other is, you can generate gradients in surface tension by using gradients in temperature, gradients in concentration, electric field, or playing with the wettability of the surface, making a hydrophobic hydrophilic patch, and a combination of all these effects. But these are essentially not the primary effect. The primary effect is the surface tension itself rather than the gradients, and that we will learn through some of the basic facets of surface tension. So, first is Young Laplace equation. So, look into this figure. So, here there is a membrane which is at configuration 1. By virtue of a pressure difference across this membrane attains a configuration 2, where the dimension of one side x, this was x, now this has become x plus delta x, and the lateral side, the other side which was having y dimension, it has become y plus delta y. The radius of curvature of this surface, this curve is r1, and the radius of curvature of this surface is r2. Okay. So, now you can see that we have a membrane, this membrane is stretched because of the pressure difference, and once the membrane is stretched, it by virtue of the pressure difference there is a work done, and this work actually supplies the necessary surface energy for allowing it to stretch. That is the basic physics that is taking place. So, there is a work energy interaction. So, what is the work energy interaction? You look into this equation. So, the pressure difference, if it is delta P across the two sides, then delta P into x into y is the force, right. If delta P is the pressure difference, that times the area, x into y is the force, times the displacement delta z, that is the work, and the surface energy is surface tension times the change in area. So, the change in area is x plus delta x into y plus delta y minus xy, and that becomes x delta y plus y delta x. Now, from similarity, geometric similarity, you can write r1 divided by r1 plus delta z is equal to x by x plus delta x. So, that means r1 is delta z into x by delta x. Similarly, r2 from the same geometric principle, it is delta z into y by delta y. So, here in this equation, if you divide it by x y delta z and then use this expression of r1 and r2, you will simply get delta p is equal to sigma into 1 by r1 plus 1 by r2, where sigma is the surface tension coefficient. So, what does this equation relate to? Of course, this equation is a very famous equation, again in elementary physics most of you have studied this equation. This is called as Young Laplace equation. This equation essentially relates the pressure difference across the two sides of an interface with the surface tension and the radius of curvature. So, the question is, what will be the pressure difference if the interface is flat? If the interface is flat, then radius of curvature is what? Radius of curvature is infinity. So, 1 by r1 and 1 by r2 in this equation will become 0. So, delta p will be equal to 0. So, on the other hand, for a spherical droplet, r1 and r2 will be equal to r. So, delta p equal to 2 sigma by r. Okay. So, this is the Young Laplace equation. There is another very famous equation called as Young's equation, and this equation defines certain parameters, and one of the very important parameter is the contact angle. So, to understand what is a contact angle, let us refer to this figure. So, in this figure you have interface. So, this is liquid, on the other side this is vapor, and the surface is a solid surface. So, when we say surface tension, actually it is a property between two phases. So, when we write the surface tension here at the solid, between the solid and the liquid, it is sigma SL. So, the surface tension coefficient for a solid liquid interface. Similarly, for solid vapor it is sigma SV, and for liquid vapor it is sigma LV, and this angle between these two is defined as the contact angle theta. So, for equilibrium the forces must be balanced in the horizontal direction, otherwise equilibrium is disturbed. So, you can write sigma LV cos theta plus sigma SL minus sigma SV equal to 0. It is a simple force balance equation, and that will give you this Young's equation cos theta in terms of the surface tension coefficients. So, of course, this is a very simple derivation, but you can give a more rigorous derivation by using energy minimization principle, but I am not going to get into that, because our objective in the biomicrofluidics is not to enter into all the possible depths of microfluidics. If you want to get into that, please refer to my NPTEL video lectures on microfluidics, but here in a nutshell we will try to understand the basics and very quickly get into biological applications, which are the prime focus of this biomicrofluidics. So, if you look into this slide, you will see that we are writing a force balance in the horizontal direction. What about the vertical direction? In the vertical direction it appears that there is an unbalanced force, but that force is balanced by a normal reaction from the surface. The other important point is that the Young Laplace equation and Young equation are necessary conditions for equilibrium, but are not sufficient conditions. That means even if these equations are satisfied, then also there can be a deviation from equilibrium. Okay. So, the next thing that we will learn is capillary rise, which is a very basic phenomenon, and we will try to learn that by a simple calculation. So, let us say there is a capillary tube and this is a cylindrical capillary. You have an interface like this, and this is the free surface. So, if on this side you have pressure P1 and on this side you have pressure P2, from the interfacial forces, the surface tension will be acting in this way. So, if this is P1 and this is P2, you can write P1 plus, so P1 is equal to P2 plus some pressure due to surface tension, the vertical component of this. So, we can conclude that P1 is greater than P2, right. So, we can write here is that now if the pressure here is P1, here also the pressure will be P1, and if you take an average height of this meniscus, see this is where practical engineering comes into picture. This is actually curved, but this being such a narrow tube, that curvature whatever it is, it can be represented effectively by an equivalent flat length. So, if that is h, then you can write P1 is equal to P2 plus h rho g. Okay. Now, by the Young Laplace equation, you can write P2 minus P1 minus P2 is equal to two sigma by r. So, this r, so let us put a different r here, this let us call it as r1. So, what is this r? So, if we assume this to be a spherical interface, see this is small. So, assuming it to be a part of a sphere is not a very wrong assumption. So, if you consider this to be a part of a sphere, then P1 minus P2 is 2 sigma by r1, where r1 is the radius of the sphere. So that is so r1 is not the radius of the capillary tube, radius of the capillary tube is r, and if this is the contact angle theta, so you can write from the right angle triangle that r1 is equal to r by cos theta. So P1 minus P2 is equal to 2 sigma cos theta by r. So 2 sigma cos theta by r is equal to h rho g. So that means h is equal to 2 sigma cos theta by rho g r. Okay. So this h is called as capillary rise. See, this rise is a generic term, it could also be fall, it depends on theta. If cos theta is less than if cos theta is greater than 0, it is rise, if cos theta is less than 0, it is fall. So cos theta, whether it is greater than 0 or less than 0, it depends on whether adhesive forces are overcoming the cohesive forces or cohesive forces are overcoming the adhesive forces. So the most important aspect of this scaling law is h is proportional to 1 by r. That means again, if you have a 1 meter diameter pipe and if you convert it to a 1 micron capillary, then the capillary rise becomes 10 to the power 6 times. So in a pipe there is surface tension, but that is not manifested as an effective force to drive the flow, but in a narrow capillary that can be an effective force to make the fluid move from one height to the other. So this capillary rise is important as a formula, but it is also important to understand that this is just the description of statics, but how fast or how quickly this capillary is being filled, that is called as capillary filling dynamics. And why it is important in biomicrofluidics is because you may have let us say a micro needle, or you may have a medical diagnostic device through which a blood sample is moving. There is a reaction site or there is a target site to which the blood will reach. Faster the blood will reach, more rapid will be the performance of your device. So to understand, first of all, how surface tension drives the blood flow in your device and how quickly does it take the blood sample from one point to the other, it is important to understand the dynamics. So we will look into a very simple model of understanding the dynamics, and that is called as a Lucas-Washburn model. So in this model, what we will simply assume is that when the fluid is moving, we are considering it as a lumped mass, and therefore it is just like a point mass moving, and it is acted upon by two forces which are viscous and surface tension, and they are balancing each other. So we will not consider gravity as such, and in this model, therefore we are considering either a horizontal channel, or even if the channel is not horizontal, because it is a micro scale phenomenon, the volumetric effects are much less than surface effects. So we are neglecting the gravity effect altogether. So the forces which are balancing are surface tension and viscous forces. See, here we have not considered viscous forces because it is a statics problem. Viscous forces come into picture when there is a relative motion between the fluid and the various fluid layers or fluid and the solid boundary. So, surface tension plus viscous equal to 0. So, how will you calculate the surface tension force? So, I will show you something very interesting. So, you multiply both sides of this equation by pi r square. So, what does it become? Pi r square h rho g. What is pi r square h? It is the volume of this fluid, that times rho into g is the weight of this fluid, right. So, this is the weight of the liquid column and this is sustained by the surface tension force. So, what is the surface tension force? So, that is the left hand side. So, that is 2 sigma into 2 pi r cos theta. So, for any geometry it is sigma into perimeter of the cross section, which is 2 pi r into cos theta. So, F surface tension we can write how will we write the viscous force? So, for viscous force we will assume that it is a fully developed flow through a circular tube and we will use the Hagen-Poiseuille's equation. So, for the viscous force, so let us draw the channel here because we will write some equations below it. So, if we write if the flow is taking place in this direction, then the viscous force is tau into 2 pi r into the instantaneous length L up to which the capillary is filled. Tau is the shear stress times 2 pi r is the 2 pi r L is the surface over which the shear stress is acting. So, this is the total shear force. So, tau, this is tau where? Tau at the wall. So, tau at the wall is what? Minus mu dVz dEr at small r equal to capital R, which is the wall. So, Vz by V average is equal to 2 into 1 minus r square by this formula we derived in the previous lecture. So, dVz dr is equal to minus, this becomes 4 V average small r by r square. So, dVz dr at small r is equal to capital R is 4 V minus 4 V average by r. So, this becomes this total expression it becomes 4 into 2 8 mu 8, there is a pi here, mu V average into L. This division by R and multiplied by R, they get cancelled out. So, F viscous will be minus 8 pi mu V L. Okay. So, why it is minus? The reason is very straightforward. It is minus because the viscous force resists the surface tension force here. So, the surface tension force will try to drive the flow along the capillary, the viscous force will try to resist the flow. There are cases when surface tension actually hinders the flow instead of driving the flow, but that is not this kind of contact angle, there the contact angle is greater than 90 degree. So, here in this diagram we have shown contact angle less than 90 degree. So, here the capillary action is driving the flow. So, now just a small trick, here in place of V average we can write dl dt. V is the rate of change of displacement is the velocity. So, you have 8 pi mu L dl dt is equal to sigma into 2 pi r cos theta. Okay. So, from here you can write L dl dt is equal to some k, where k is sigma 2 pi r cos theta divided by 8 pi mu. So, if you integrate it, you have L square by 2 is equal to k dt, sorry, k kt, not k integral dt plus some constant C1. So, at t equal to 0, if L equal to 0, then you can see that this C1 will be 0, and then you get a very interesting relationship between L and t. You will get L is proportional to square root of t. Okay. So, this is the hallmark of the Lucas-Washburn model. I could have discussed about many other models given an opportunity of time, which you can look into my detailed NPTEL lecture on microfluidics, but I feel that the Lucas-Washburn model is the most fundamental model, because it captures the essential force balance, and you will see that no matter what kind of fluid it is, you can cast it in this very simple model. So, if for example instead of water, blood is filling into the capillary, then what will happen to this model? So, what will happen to this model if blood is filling the capillary is the fact that instead of this expression for velocity profile, there will be a different expression for velocity profile, and not only that, the wall shear stress, instead of using the Newton's law of viscosity, it will be something else. So, this viscous force mathematical expression will change, but the physics, the concept still remains the same. Surface tension and viscous together they make a net zero force in a inertia free regime. So, with this understanding, I will try to give you a case study on capillary dynamics of blood flow through microfluidic channels. This is one of the very interesting case studies in biomicrofluidics. So, if you look into this equation, so now I will show you an equation where, so you have a surface tension force, you have a viscous drag force, and you also have an inertial effect. So, this inertial effect is kept here with an understanding that this is a more general model than the Lucas-Washburn model. The Lucas-Washburn model, the left hand side, that is the ddt of mv, this is essentially the Newton's second law. So, the left hand side is ddt of mv, rate of change of linear momentum, and right hand side is force. In the Lucas-Washburn equation, this term is taken as 0. So, that is called as inertia free regime. But if inertial effects are there, so in the mass you will have a fluid mass that is ready to enter the capillary, which is called as added mass. Added mass is the, so if you for example consider that this is a capillary and some fluid is entering into it. So, this additional mass which is outside the capillary but an integral part of the system, so that is called as an added mass. So, on the top of the added mass you have this mass within the capillary. So, this total mass ddt of mv is the resultant force. The shear stress depends on how you model blood, and here I will try to describe a little bit of hematological issues that can crop into picture when we are modeling blood. So, you will see that blood can be modeled in various ways, like there are, blood is of course intrinsically a viscoelastic type of material where it is partly viscous and partly elastic because of the blood cells which are there inside. So, the blood constitutive model, if you look into this equation, the shear stress, this is the stress tensor so called, it is k into the rate of deformation tensor to the power n. So, this is a power law model. There are other models like Casson model, there are different types of models. So, if you have this power law model, this k and n, these are not constants, but these are functions of certain parameters, and those parameters are considered within this C1, C2, C3. These are constants, but constants for certain individuals, these are different constants, and then on the top of that you have a very important parameter in hematology which is called as hematocrit. So, hematocrit is the volume fraction of the red blood cells or the packed cell volume. So, hematocrit or the packed cell volume is a very important hematological parameter, and you will see that this is commonly used even to monitor certain diseases. For example, after dengue fever, people start monitoring not just the platelet count, but also the packed cell volume or the hematocrit. So, blood essentially, blood is a very complex fluid, and that is why there is a great challenge in modeling blood, but blood fundamentally is a collection of cells in plasma, but at the same time we have to understand that is plasma just like water? Plasma is not just like water, because plasma contains certain proteins. These proteins are primarily albumin, globulin and fibrinogen, and these proteins form their own network type of structure, and that makes the rheology not exactly the same as that of water, although it looks like water. So, the blood contains primarily the red blood cells, the white blood cells and the platelets. These are the three different types of cells, and you can see in this view graph the shape of the red blood cells, and these are like biconcave discs. So, under a shear this biconcave disc shape is disrupted, and there is a shape variation of the red blood cells, and that can alter the dynamics very interestingly, and therefore the rheology of blood is that its shear is altering the effective viscosity. Effective viscosity is also a function of shear in the blood. So, unlike the red blood cell which is commonly affecting the flow dynamics, so if somebody asks you that what aspect of the blood affects the dynamics of flow, so the most important aspect is the inclusion of the red blood cells, but the other factors are also important, like the white blood cells which protect the body from diseases, and platelets which are not normally influencing the flow, but platelets are important in forming blood clots, and they may severely interfere with the flow if blood clot formation is either enhanced or resisted. So, platelets may not be directly influencing the rheology, but in case the platelet count goes below a certain limit or beyond a certain limit, I mean these can be affecting the fluid dynamics to a significant extent. So, as I told you, now all these factors, they come into these parameters C1, C2 and C3, which I have mentioned in this slide. So, this C1, C2 and C3. So, C1 depends on the plasma globulin concentration and hematocrit. C1 increases with a parameter which is called as TPMA, total protein minus the albumin fraction. Similarly, C2 also increases directly with TPMA. C3 is a relatively independent parameter in a sense that it does not depend so much on the plasma chemistry and appears to be remarkably constant for a given animal species. And for a given blood sample, how will you know what are C1, C2, C3? That is a big question, and these these depend on the training of your model with a given data set, from a large number of patient samples, so that your model knows that what are the parameters based on which C1, C2 and C3 depend, and accordingly they can fit the model. So, C1, C2, C3 for a given blood sample depend on chemistry of plasma, chemistry of RBC, specifically its hemoglobin concentration, number of the red blood cells, their size and degree of cellular aggregation, the red blood cell shape, geometry and deformability, mass density of the red blood cell, plasma and composite whole blood. So, you will see that these appear to be parameters, but these parameters are so strongly dependent on the composition of the blood. Now, hydrodynamics of blood as it is flowing through a microfluidic channel, it may be affected by certain diseased conditions, but even without that, a very important aspect which I want to highlight is the Farius-Lindquist effect. So, what is this effect? If you look into this graph, the top graph in the figure, you will see that as you reduce the diameter of the blood vessel, the apparent viscosity, instead of increasing, it actually reduces. So, in a very small channel, how blood can remarkably flow by surface tension is because of the following two things. One is that if the channel is very narrow, then surface tension drives the blood flow in a remarkable way. The second is that if the channel is very narrow, the viscous effects also become less. And why the viscous effects become less is because at the wall the red blood cells form a cell free layer. So, cell free layer means actually the red blood cells come out of the wall and join the center line. So, the wall of the blood vessel, the wall of the microchannel, this wall becomes devoid of the red blood cells. Had the red blood cells being placed at the wall, that would have enhanced the viscous effect, but now the walls are devoid of the red blood cells, and that makes the situation such that the effective viscosity is less. So, this in medical science is a very remarkable phenomenon and called as Farius-Lindquist effect. So, with all these things, it is possible to make a mathematical model which shows that how in a microfluidic channel you can transmit blood by using surface tension. So, if we know this becomes one of the basic premises of designing microneedles and designing medical diagnostic devices, which primarily work on capillary action. For example, medical diagnostic devices on paper substrates on which capillary action only drives the sample. So, if you are interested to learn about the details of the mathematical modeling, I have referred to a couple of my papers. One is published in Lab on a Chip and another is in Analytica Chimica Acta. These are published in 2005 and 2007, and I have given the citations of these two papers, and I believe that if you go through this, you will get an insight on how capillary transport of blood in a microfluidic channel can be modeled in a very simple way. So, with this, let us draw a conclusion to this particular lecture. So, in this lecture we have studied what is surface tension, what is capillary rise, and how surface tension can drive a flow in a microchannel, and how that modeling can be enhanced in terms of physical and mathematical understanding when it is blood instead of a Newtonian fluid. Thank you very much.